Well, I'm going to really start where Matthew left off. So this idea of information. And Francis Crick his ideas really went right to the bottom of evolutionary biology as well. So it wasn't only the code itself that he was talking about, and here again we see the terms information keep coming up, but this was 1958, and he said, biologists should realize that before long we shall have a subject which might be called protein taxonomy. Now, we'd talk about DNA rather than proteins now. The study of amino acid sequences of proteins of an organism and the comparison of them between species. It can be argued that these sequences are the most delicate expression possible of the phenotype of an organism, and that vast amounts of evolutionary information may be hidden away within them. So it's quite a well-known phrase now, but this was really the beginnings of phylogenetics as we know it. Or the gene which Matthew showed at the very beginning, long sequences of letters, we now simply compare these sequences of letters in different organisms. Now, the first person to actually do that was Linus Pauling, and again, it's very striking to me. Linus Pauling, of course, was a physical chemist, Crick, a physicist by background, and these are the basic ideas underpinning really the last half century of research in evolutionary biology. So what Linus Pauling did with Emil Zuckerkandl in 1962 was they started comparing different proteins, the number of different amino acids in proteins from different places. So they compared hemoglobin, the beta chain of hemoglobin, the gorilla, the horse, and the human, and they found there's 20 different amino acids if you compare a horse with a human being. There's only one different amino acid if you compare the same protein in a gorilla with a human being. So they also introduced this idea of a molecular clock, a rate of replacement of amino acids over long periods of evolutionary time. And from fossil evidence, they figured out that horses and humans must have diverged about 160 million years ago, something like that, and therefore the rate of change was something in the order of there should be 10 amino acid changes over that period. So it will be around about 12 million years per amino acid that changed, or thereabouts. So this was the beginnings of phylogenetics. But hemoglobin, of course, is not conserved by everything. If you really want to try and get back to the origins of life on Earth, then you need to find proteins or genes by this time which are shared by absolutely every living thing. And the one thing which every living thing really does is make proteins. As Matthew was talking about, you've used the DNA as the code. You produce an RNA transcript from that, and that transcript then is used to make proteins on the ribosomes. And all life has ribosomes, and those ribosomes are made up of various, effectively, machine parts. And one of those machine parts is this. So this is the small subunit ribosomal RNA, and Carl Woese picked on that back in the late 1960s and started to compare the structures of these. So this is yeast. The human one is very similar to that. This is E. coli. You can see these molecules are remarkably similar right across all of life. And so what Woese did was to compare these molecules, and he came up with this tree of life. And this is probably familiar to most people. How many people are familiar with this three domains tree of life? Could we have a show of hands? So probably about half or more than half the audience by the look of it. It's in all the textbooks, and it's wrong, actually, and I'll explain why. But it was shocking at the time, and it's still important. And now, these are the bacteria. So we're all familiar with bacteria. Again, the archaea, probably if you're familiar with this tree, you'll be familiar with the archaea. They look exactly the same as bacteria. You cannot tell the difference. We've actually known about some of them, methanogens, for 400 years. They were first discovered by Volta, of all people. But they look exactly the same. We had no idea that they were different. But you can see from the length of the branches on here, that gives an indication of the number of different sequence changes between them. And so the longer the branch, the more sequence changes there are. And so it gives a sense of time and evolution and experimentation over evolutionary time. So here we are. These are the animals here, the fungi, plants, and so on. Very little variation there compared with this enormous amount of variation going on in these two other groups. So this is quite a shock. There's an entire group that nobody knew existed. And then we're consigned to a small corner of the tree, which is pretty trivial. And, is that really what the world looks like? Well, people have been sequencing these genes ever since, of course, and this is ribosomal RNA again. And just to give you a sense, this is where we are. Now, this is something which I think is really important in modern science. Is the amount of information in here is absolutely massive. Does it mean anything? Well, not a lot, actually. And the reason you can immediately see, so animals are up here. And I can't actually read my own slides, but the plants are up there, the fungi are down here. Look at this little bit. The bacteria and the archaea are down there. They're a vanishingly small part of that tree. So already something's going on here. We're actually obsessed with animals and plants, and we sequence them all. And probably they've not included everything on here anyway. But if we go back, it's a very different picture to this. So, and this is one gene. Now, the question that I think the big question that I'm interested in here is what on earth was happening down here? So these groups had essentially 4 billion years of evolution, going right back to 4 billion years ago, and effectively infinite population sizes, and that's what they looked like. What have they achieved in all of that time? Almost nothing. It's shocking. They're in their biochemistry, they're amazingly complex, but in terms of their appearance, their structure, their morphology, they're shockingly simple. Why didn't they get any further? What was happening down here that wasn't happening over there? Why is it that Carl Woese came up in that group, but why can't we have a bacterial human? Why doesn't that happen? Well, it's not only at the level of large multicellular organisms. This is really written deep into the structure of cells themselves. So this is a bog standard alga. You'll find that in any pond, Euglena. This is a relatively complex bacterium. It's even got a little compartment that some people have said maybe that's a nucleus. It's not, but the reason you can't see it very well is this is roughly to scale. And so this is a single-celled alga. When you get a whole lot of them, they form a scum. And you don't need to know what any of these things are. This is the nucleus. These are chloroplasts, in fact. But you don't need to know what any of these things are to appreciate that there's an awful lot going on and it's very big. And the same goes for the number of genes. They have, 3,000 families of new genes seem to have arisen with just this group. So, and they can have genome sizes up to about 100,000 times larger than the largest known bacterium, so enormously larger. And this is, just to give you a sense that it's actually quite hard. Any suggestions for what the cell on the left might be there, or indeed the cell on the right for that matter? Okay, so this is paramecium. Again, you'll find it in ponds. And this is a human pancreatic acinar cell. It's pretty hard to tell the difference between them, certainly in terms of their overall complexity. Now, paramecium actually has 40,000 genes. It has twice as many genes as humans have. It's a large, complex organism, but it's a single cell. And so my point is that eukaryotes, this group of complex cells, are just enormously more complex than bacteria or archaea. Something happened, and we don't really know what it is. So there's a paradox there, and it's a paradox about natural selection itself. So essentially, the problem is that all the complex life that we know, and that includes amoeba and paramecium and so on, is composed of this one cell type. And all those cells are essentially the same. It's very difficult to tell the difference between them. They all share a long list of hundreds of or thousands of traits in common. It only arose once. We all share a common ancestor, and that common ancestor, by definition, arose once. You could say perhaps there were lots of other origins as well, but there's no trace of it anywhere in biology. So, it's make-believe. It might have happened, but there's no evidence for it. We all share these traits. Prokaryotes, that means the bacteria and the archaea, they show essentially no sign of evolving any of those traits. They have, I showed you the nuclear-like compartment, that's about as good as it gets. So there are steps in the direction, but really nothing very impressive in comparison to what eukaryotes do. Now, if all of those traits, if the nucleus or if sex, for example, arose step by step, as natural selection says it must, and each of those steps offered some small advantage, which I'm not disputing at all, that's what happened, then why did none of them arise in bacteria or archaea? Well, we can begin to get another clue. Well, actually, before I get onto that, let me just give an indication of why this is really a paradox. So the eye, for example, eyes have arisen on at least 60 or 70 different occasions, independently, from a light-sensitive spot on some kind of an early worm. So there are some genes in common, some regulatory genes in common, but they independently recruited all the rest of the genes required to make an eye. And so these are eye flaps on blind shrimps that live in hydrothermal vents. These are scallop eyes, fantastic things. This is an eye spot in Euglena. So it's a single-celled alga that I showed you before. This is a trilobite. I won't go through the list, but they're all morphologically completely different. It's exactly what natural selection will predict. Different environments, different circumstances, you end up with different types of eye arising on multiple occasions. So why didn't that happen with sex or with the nucleus or with phagocytosis, the ability to engulf other cells? So here's the beginning of a clue. This is another tree, what we would call a star tree. This is the common ancestor of all eukaryotes. Now, you might think that eukaryotes are split up into plants and animals and fungi. I'm afraid that turns out not to be true as well. This all turns out from the information of comparing all of these genomes. So here we are, the metazoans. There are the fungi. Plants are over here somewhere. These are the supergroups of eukaryotes. Have you ever heard of chromalvulates or uniconts or rhizaria? Most biologists haven't either. I mean, these are only evolutionary biologists care, and only people who care about the evolution of eukaryotes care about these terms. But it's a very, very striking and different portrayal to anything that people had expected. And two things to note from this. One of them is that these branches here, there's very little variation. There was some kind of a big bang radiation very early on, and then there's far more variation within the groups than there is between the ancestors of the groups. The other thing is this black hole at the center is pretty symbolic, because that common ancestor of all eukaryotes had all of these traits. It had basically everything. And we don't know where they came from. So we start out with some kind of a bacterium. Let's say it's a complex one like this. There was a recent paper that people may have heard of saying there's a group of archaea called the Lokiartiota, which are perhaps some kind of missing link, but they are not. They are basically similar to this. So we start here. This is all we have evidence for, and there are no surviving evolutionary intermediates whatsoever. So there's very, very little to go on to say, well, how did the nucleus arise? Why did the nucleus arise? What about sex? What about all these things? How can we possibly get at those questions? Well, we can get at them, and again, we can get at them by looking at gene sequences. Some genes sitting in our own nucleus are very comparable to bacterial genes. If you lay out their sequences and you compare them, so the ribosome genes, for example, that I mentioned, we have them, and you can compare them with bacteria. And you can compare them. It turns out that we have large numbers of genes that are basically quite similar to bacterial genes. And we have large numbers of genes which are basically similar to archaeal genes. We are some kind of chimera. We also have a large number of genes which don't compare at all with either of those groups, but on our basis, about 40% of our genes do compare with prokaryotes. The strange thing about this is these are the groups that they are most similar to. So you take a gene, you compare the sequence, and you say, of all the bacterial genes that we have, which group is it most similar to? And it turns out to be this one, or that one, or this one. And so all this shows is all the eukaryotes, the animals, plants, fungi, and so on, we all have these bacterial genes, and we have them basically in the same way. We've not been acquiring them over time from bacteria, different bacteria at different times. They all were there from the beginning. But the trouble is they come from all these different groups. So how can we interpret that? This is a problem, the kind of problem that people wrestle with, which is to say, there are several possible explanations, and we don't know which one's true. This is a fact, but how do you wrestle with it? One possibility would be Lynn Margulis, her serial endosymbiosis hypothesis, theory. I don't like that very much, but it does make predictions. It makes predictions that it's serial for a start. You have a whole series, and actually not enough. She's got 3 or 4 of them here. People have been looking for this group for 30 or 40 years and never found a single thing from that group there. So that's why I don't like this as a theory very much. But it also says serial, and that means one after another, and they don't all arrive at the very beginning. So it doesn't really explain that. This begins to. So this is Ford Doolittle wrote a great article entitled "Taking an Axe to the Tree of Life." He really does not like the tree of life. Why? Because of lateral gene transfer. So what bacteria do is they pass genes around between themselves as if it's loose change. So they pass all their genes onto the daughter cells, but then the daughter cell grabs one from here and one from there, and everything gets mixed up. And so if you try and track back a tree of life, well, maybe you end up with a mess like that. Again, it's very hard to know. It's very hard to prove what came from where. If we make a simple assumption, which is that the common ancestor of all of life had a genome which was basically similar to a modern bacterial genome, let's say it had 4,000 genes or something, then you can constrain the amount of lateral gene transfer that probably must have happened over, and this is the picture that you get. So there is some sign of vertical inheritance, but it's a mess. I mean, just think of the ribosomal RNA tree that I showed you with this rather spurious sense of precision that everything is beautifully laid out. We know exactly how everything's related to each other. We do not. This gives a sense of the scale of the mess. But it also gives a sense of where the answer might be. So if this was actually a singular event, so one bacterium and one archaeon came together, and the reason that all those genes seem to branch with different groups is that there'd been lateral gene transfer ever since between those groups, then that explains it. This then is a hypothesis, which is testable in principle at least. It was a singular event. It wasn't multiple endosymbioses. It wasn't something happening over time. It was just once and right there. And actually, there's a lot of evidence behind that now, and I'm not going to go into it today. But it's what Jim Lake called the ring of life. And I'm told he sent a paper to Nature entitled "One Ring to Rule Them All." And unfortunately, well, they accepted the paper, but not the title, sadly. So this is Jim Lake. This is his ring of life. And there's a lot of evidence now that the host cell was an archaeon from the archaea, and there was an endosymbiont, so a cell that got inside it. And that cell went on to become the mitochondria. So we all have mitochondria, the powerhouses of eukaryotic cells. So it must have been something a little bit like this. So this is the one example that we know of today of a bacterial cell. This is actually a cyanobacterium, and you can see there's some membranes in there. These are thylakoid membranes in cyanobacteria, with cells living inside them. We have no idea how they got in there. They weren't engulfed by phagocytosis, because this is not a phagocyte. It has a cell wall. But they're there all right. We just don't know how they got there. But this is the starting point then for the evolution of complex life, if any of this is true, which it might not be. So they went on, I'm suggesting, to become mitochondria. This was the discovery of mitochondria by Richard Altman back in 1895, and he used an osmium stain which actually dissolved the cytoplasm pretty much, leaving just the mitochondria and the nucleus. He thought the nucleus was a food dump, and that these were the elementary organisms, what he called bioblasts. And so he had a great job title, Extraordinary Professor of Anatomy. Love to get a job title like that one day. Anyway, but what we know now is that these mitochondria were bacteria once, and they're amazingly successful if you think we have something in the order of 500 trillion mitochondria in our own bodies. So I mean, you will hear it said that we're only one-tenth human, that there's 10 times as many bacteria in the microbiome. Well, there's at least 500 times as many bacteria as mitochondria living in us. We are basically 40% mitochondrial. And this is what's going on in you right now. So these are mitochondria, and these membranes is where respiration is taking place. So as we're burning food in oxygen, it's all going on in these membranes inside these mitochondria, which were once bacteria. And this is roughly what's going on. So in a nutshell, we're stripping electrons from food, hydrogen really from food. The electrons are flowing down this wire of proteins. So there's a current of electrons to oxygen. So we're stripping them from food. And the protons, so hydrogen is made up of electrons and protons. The protons are pumped across the membrane. So the flow of electrons, the current of electrons flowing to oxygen, extrudes the protons across the membrane. And we end up with something which is a little bit like a hydroelectric dam. So we've got a reservoir of protons on one side and very few protons on the other side. And then we have a flow of protons through the ATP synthase, and that produces what's called the universal energy currency, ATP. So it's a very peculiar way of going about things. Nobody guessed that that was how life was going to work. Now, those little blobs that I showed as the current of electrons, actually, they're amazingly complicated proteins. It's been a triumph of research over the last 30 or 40, 50 years to work out the exact structure down to practically the scale of atoms now of, well, this is the ATP synthase, these are the respiratory complexes that are pumping protons across the membrane. So we know in enormous detail how these things work. There's still some mysteries, there's still people actively working on it, but we know an awful lot. What we don't know is why life works that way. That was the problem that Peter Mitchell, who came up with this idea in the first place, it was what motivated him. Why, not how does respiration work actually, but how do bacteria keep their outsides different to their insides? It's one of these very simple, very deep questions. How do they keep different to the outside world? And if you can keep different to the outside world, he realized that, well, stuff can flow in again and you can take advantage of that. And so the flux of things into cells give you a source of energy. So this is Peter Mitchell and Jennifer Moyle. So Jennifer Moyle did all the experiments. Peter Mitchell did all the thinking. He got the Nobel Prize. She didn't. We can debate the pros and cons of that later on, but basically it was her experiments that proved it, but it was Mitchell who really conceived the ideas. Who's to say what's most important in science? Why? I'm not going to talk, there have been several leads to say I'm going to talk about the origin of life. I'm afraid I'm not. You're going to have to read my book. I'm just going to say a word or two about it. How did these things start? They're universal across all of life. I failed to quote this lovely quote from Leslie Orgel. Not since Darwin has biology come up with an idea as counterintuitive as those of, say, Einstein, Heisenberg, and Schrödinger. The physicists are creeping back in again here. But physicists are full of big counterintuitive ideas. There aren't very many in biology. The double helix is an entirely intuitive idea. Jim Watson said, it was so pretty it had to be true. But the idea of pumping protons across a membrane, it's so ugly that you think, why on earth does it work like that? And nobody can ever remember the term chemiosmotic coupling. So the problem is that these proton gradients, they're as universal as the genetic code. Now, there's a sleight of hand in there because Matthew said that the genetic code is not actually quite universal, and it's also true that proton gradients are not quite universal either, but they're more or less universal, and the groups that don't use them are obviously relatively recently derived and live in fairly peculiar conditions. So in a broad brushstroke, they are universal. And that implies they arose really early, and that means that we need to think about what kind of an environment. It gives a pointer to what kind of an environment life might have arisen in. And I'm going to skip over this very quickly. These kind of vents, which were discovered only 15 years ago, called alkaline hydrothermal vents, down at the bottom of the Atlantic, seems that they're probably far more common, or would have been far more common 4 billion years ago. Mike Russell, who put these ideas forward over the last 20 years or more, has argued that they're electrochemical flow reactors. So it's not like a black smoker belching smoke out of the top. It's a labyrinth of pores, and the hydrothermal fluids wend their way through these pores. So the important point for us is that they have natural proton gradients. These are alkaline fluids going into what would have been a mildly acidic ocean. And so the inside has very few protons, the outside has lots of protons. And they would have been across these barriers in this microporous labyrinth. And so this is the situation that we have in these vents. We've got large concentrations of protons on the outside, low concentrations on the inside, and some kind of thin inorganic barrier between the two. This is what's going on in all of life. So this is a bacterium or something. It's the same thing. It's pumping protons out. It's actively pumping them out. This is a thin membrane. This is orders of magnitude thinner than that. So I'm just exaggerating the analogy here. But it's about the same order, which is to say it's about 3 or 4 pH units different between here and there. Now, there's all kinds of reasons that the question is really, what use is that without proteins, without membranes? I do discuss that in the book, and I'm not going to talk about it now. But for the last minute or two, few minutes, I want to suggest that the problem with this is that they are constrained by pumping across their membrane. This is how life started. Bacteria, archaea, all do that in that way. And then they're kind of stuck with it. So what was the good thing about having this situation at the origin of eukaryotes, where you have an endosymbiont? This goes on to become the mitochondria. Well, the thing is that these are cells. It's not just a cell, it's a population of cells, and they can compete among themselves. And when they do so, very often, if you can make more copies of yourself in a given period of time, you will be better represented down in history. And so if you get rid of a few genes that you don't need, for example, you make more copies of yourself in a shorter period of time. And so the competition between these cells in this population living within a cell, they tend to dump any genes that they don't need and become simpler and rely on the host cell for anything that they can do. And in the end, they lost almost all of their genes. We only have 38 of them left in our own mitochondria. Well, there are massive, massive energetic consequences of losing those genes. So just as a quick and slightly silly thought experiment, imagine that you've got a cell and it's got 100 endosymbionts. So it's got 100 bacteria living inside it. And let's say that it has a standard bacterial genome. So it's got something like 4,000 genes, 4 megabases, 4,000 genes. It loses 5% of them. So these are the genes it normally would use for making a cell wall to protect itself. It doesn't need that anymore, and they replicate a bit faster if they lose those genes. So it's lost 5% of its genes. So what's the energy savings that you get from losing those genes from 100 endosymbionts? Well, each of those genes encodes a protein, and those proteins are produced in multiple copies. You don't just make one protein, you make thousands of them. On average, in a bacterium, about 2,000 of them. And each protein is a string of amino acids which are all linked together. On average, 250 amino acids linked together in a protein. And it costs you about 5 ATPs to bind the two amino acids together. So you can work out the cost in ATPs, this energy currency, that you would have to pay to build all of those proteins? Well, the answer is the cost is something in the order of 50 billion ATPs. If we assume a 24-hour life cycle, that works out at 580,000 per second. So that's what you would save by not expressing those genes in those 100 endosymbionts. What could you spend it on? Well, let's have a dynamic cytoskeleton, something that will allow you to move around. What does that actually cost? There's no reason why you should spend it on that, but you could spend it on that, so what would it cost? Well, the length of the actin, this is made of a filament, but the filament is lots of proteins joined together, they kind of gum together. And each of those monomers is 29 nanometers in length. So there's the 35 monomers per micron, so that's 35 proteins joined together per micron. There's 374 amino acids in each protein. It's a dimer, and again, we're assuming 5 ATPs per peptide bond. So the total cost is 131,000 ATPs to make 1 micron of actin, which means for those savings, you could make 4 microns a second, which is a massive amount. That's way more than any bacterium actually does. And that's de novo synthesis starting from the start. So it really is it's a huge difference. Now, mitochondria didn't lose 5% of their genes. They lost 99% of their genes. And they're not limited to 100 in a cell. There can be as many as 300,000 in a large amoeba. And so the actual energetic advantage that eukaryotes have as a result of gene loss in their endosymbionts, which have always retained a few to keep control over respiration, it's absolutely huge. And I think that's what made that difference. I mean, we can quantify that. These are various different types of bacteria, including giant bacteria. There are some giant bacteria which are visible to the naked eye. They're still only 0.5 millimeter long, but that's pretty big for a bacterium. They have what's called extreme polyploidy, thousands of copies of their genome. If you take all of that into consideration, these are various numbers for bacteria. This is the metabolic rate, so the power that they have per gene. And these are eukaryotes. This is a log scale, so this is 3 or 4 orders of magnitude more energy per gene. This is Euglena again, which I was showing you earlier on. This is a large amoeba. So orders of magnitude more energy to do what you want with your genome, to make that many more proteins or to have that many more genes. So just for the last bit then, coming back to this, can we get any insight into what was going on down here? Well, possibly, because if this is just a complex bacterium, but it didn't evolve by natural selection in a large population, what actually started out is something more like that, then we can begin to constrain what was going on. And we've been doing some population genetics on this in the department. Zina Hadjivassiliou just finished a PhD about a year ago working on this. I'm not going to talk about that now, but you can see immediately just looking at this that if you have a population of mitochondria within a cell, they differ. They mutate in different ways. They can become different. They segregate in different ways. And it takes quite a lot of work to try and figure out how do the organisms constrain what's going on here. In fact, the mechanisms are almost certainly things like having two sexes, having sex at all, having a germline. All of these can be shown that they actually improve the quality of mitochondria. So I'm not going to say more about that. Again, if you're interested, you'll have to read the book. But right up to the Cambrian explosion and the origin of animals and germlines and so on, it's all potentially explicable. As always with science, these are neat ideas. They may not be true, they may be complete rubbish, but at least we can test them, or we can try and find ways of testing them. So, a few conclusions. Complex cells, the eukaryotic cell arose just once in 4 billion years. That seems to be a fact. I can say more about that if people are interested. It looks as if energy constraints on bacteria demand some kind of a rare endosymbiosis between prokaryotes. Conflict then between the endosymbionts, between the cells living inside a host cell, that's pretty tough, and it forces cells down particular populations of cells down particular avenues. It forces you to have sex, for example, or to have two sexes. And so we can begin to predict that the resolution to those conflicts may account for why all eukaryotes share so many similar traits. Now, I want to leave you with this. This was discovered, in case you think it's pessimistic that a complex life only arose once in 4 billion years, maybe it's happening again off the coast of Japan. So this was discovered about 5 years ago, and it's an intriguing title on the paper, A Unique Organism from the Deep Sea. Prokaryote or eukaryote? They don't know. I don't know. Nobody really knows, but let's have a quick look at it. So it's about 10 microns long, which is substantially larger. These are bacteria here, so it's substantially larger than the average bacterium. It has what looks like a nucleus, so this looks like a standard eukaryotic nucleus. It's got some endosymbionts. Perhaps they're not mitochondria, but they could be something like hydrogenosomes, which are derived from mitochondria. It's got a plasma membrane and a cell wall and so on. So to a first glance, this looks like a standard eukaryote. But when you look more closely, well, this is not a double membrane. All eukaryotes have a double membrane around the nucleus. This has a single membrane around the nucleus. All eukaryotes have chromosomes with a diameter of around about 30 nanometers. This has bacterial filaments with a diameter of 2 nanometers. We don't know what those are. There's no genome sequence for this. We really don't know very much. This mottling here, those are ribosomes. No eukaryotic cell that we know of has ribosomes in the nucleus. The whole point of the nucleus is to exclude protein synthesis from going on there, but here it's going on in the nucleus, as well as outside the nucleus. These membranes inside the cell, they are nothing like the membranes that we see, the endoplasmic reticulum, things like that. So this is not a eukaryote, and it's not a prokaryote. I would say that what this is a bacterium that acquired endosymbionts and which is recapitulating eukaryotic evolution. It's become larger, it's accumulating genes, it's got a nucleus, it's going through the whole thing all over again. But we only ever found one of them, and they've been looking for 15 years to try and find another one, and they've not found it yet. So, and they took a section of that, which means we don't have a genome sequence either. So this is how science goes. We're just going to have to wait, and you are as well. Thank you very much.
What is the symbiotic relationship the two organisms have that allows them to be symbionts?